Welcome to Study Loop.
So how does the presence and absence of glucose and lactose affect the lac operon? So first, let's take a look at what the lac operon is. It is a gene. So let's draw the gene here. And right before the gene, you have an operator and a promoter, which is basically the CAP, and that's the CAP binding site, and that's where the CAP binding that's where the CAP protein binds to. Okay, it's going to become clear in a second, I promise. Just stay with me here. And this is the operator. And drawn in black is the actual gene that needs to be transcribed. Now, the roles of the cap binding site and the operator are that to the operator, a repressor can bind. So this is a repressor. And to the CAP binding site, a CAP molecule can bind, or CAP protein. Okay. And the CAP, it's called the CAP protein because it binds cyclic AMP. And cyclic AMP, which I drew in this small triangle here, this guy right here, so cyclic AMP is what activates the CAP protein to actually bind to the CAP binding site. And once that happens, the CAP promotes the binding of polymerase to this guy. So you need two conditions, okay? So in this instance, so the example that I've drawn here, this would be not functional. Because you have the repressor on. In order for the operon, by the way, it is the entire gene plus operator plus the CAP binding site. The entire segment is called the operon. Okay, so in order for the operon to be on, to be functional, you need. So let me draw the DNA here. So that's the gene. This is the operon, and this is the CAP binding site. You would need the repressor to not be on, but you need the CAP molecule to be on, of course, with the cyclic AMP that activates it. And this then would have the RNA polymerase in there, and the gene would be transcribed. Okay? And whether the repressor is on or off, and whether the CAP protein is on or off, are regulated by the presence or absence of lactose in the cell. Now, why does this matter? What I have here on the right, this is lactose. I'm going to abbreviate here for the sake of time. And this is glucose. Now, you see glucose on the left here, it's a monosaccharide, whereas lactose is a disaccharide. Lactose, one of the components of lactose, is actually glucose. And the reason that this matters is that in order for a bacterium to be able to consume lactose, it first needs to break this bond right here, whereas it doesn't need to go through that step with glucose. So then if glucose is present, you want the operon to be off because you want to prioritize the consumption of glucose. You want to turn the operon on when you don't have glucose and when you have lactose. Okay, so this is what that looks like. So up here, let me switch to the pen. Up here, you have the entire operon right here. You don't have the cyclic AMP with the cap protein, you don't have that on, and you don't have the repressor on as well. So this is how it happens. Let me just do this. Glucose, it represses the production of CAMP. And this guy promotes transcription. So when the CAMP and the CAP protein together are on the operon, that means that there's low glucose. So this would be glucose minus over here in this guy right here and right here. Okay, you would have glucose present here because the CAP is not on, and over here you would also have glucose present. Okay, and lactose prevents the repressor from binding. So if the repressor cannot bind, then the polymerase can move forward. If the repressor binds, then it cannot move forward. So here you have glucose present and you have lactose absent. So the cell has no reason to turn the operon off. It has plenty of glucose. Over here you have glucose not present and lactose available because the repressor is not on. So this is the functional one right here up top. This is the functional scenario. Okay, so that's kind of how the presence of lactose and glucose work. Glucose acts as preventing the promotion, and lactose acts as removing the repression. And it's this two-factor control that makes it possible for a bacterium like E. coli to only turn this up because it is a metabolically expensive process, right? So it only turns this on when glucose is present and lactose is absent in order to prioritize the consumption of glucose because that's a much easier food source to consume.